Mankind has witnessed countless pandemics like the bubonic plague, the Spanish influenza, and the current COVID-19 pandemic. These pandemics usually lasted for years until the majority of the population developed herd immunity from the infection. However, passively waiting for this natural immunity to develop is not a practical solution, as this will cause innumerable unnecessary deaths. With the development of vaccines, the human immune system can be taught and armed to fight off the agents of diseases that lead to pandemics. Vaccines are products that contain a weakened, inactive, or altered form of the disease-causing pathogen that, when introduced to humans, can incite the production of antibodies. So, with this, one may gain immunity without actually acquiring the disease. Over the years, several types of vaccines have emanated but are not limited to the following. First, the live attenuated vaccine is composed of live pathogens that are weakened and tamed by passing the disease-causing virus through a series of cell cultures or animal embryos. Second, we have inactive vaccines wherein the pathogens are made inept to replicate by employing heat or chemicals such as formaldehyde and formalin. Subunit vaccines utilize a single part of the disease-causing pathogen, the antigen, by isolating the specific protein from the bacterial or viral pathogen. Toxoid vaccines combat the toxins the bacterium produces, but not the bacterium itself. Lastly, the mRNA vaccines is a type of vaccine that does not contain live pathogens, but delivers genetic instructions to the cells to make a viral or a bacterial protein to trigger the adaptive immune system. But how do vaccines work? Each pathogen contains a distinct antigen. An antigen is a molecular marker that triggers the adaptive immune system. The three main objectives of the adaptive immune system are to detect, to attack, and to remember. As soon as the vaccine enters the body, the dendritic cells will engulf the weakened pathogen via phagocytosis. During the process, the antigen will be presented on the surface of the dendritic cells with the aid of the MHC class II molecules, thus activating the adaptive immune system's response. The helper T cells will then act as the key mediators to keep the process going. They will pick the antigens up from the dendritic cells to pass them on to the B cells. Next, the B lymphocytes will then be converted into plasma cells and will start manufacturing antibodies. Soon, these antibodies will bind themselves to their complementary antigen. And to fully eradicate the virus, the phagocytes will destroy the pathogens by ingesting dead or dying cells, while killer T cells, on the other hand, will detect and destroy infected cells. The adaptive immune system will soon develop a memory in which it is ready, in any circumstance, to produce the correct antibodies if the same pathogen shows up once again. Let's go!